So we are back from the break, and our first speaker is going to be Ed Boyden. He's a professor of neurotechnology at MIT, an investigator of the Howard Hughes Medical Institute and the MIT McGovern Institute, and a professor of brain and cognitive sciences, media arts and sciences, and biological engineering at MIT. He leads the Synthetic Neurobiology Group, which develops tools for analyzing and repairing complex biological systems, such as the brain, and applies them systematically to reveal ground truth principle of biological function, as well as repair this system. He received numerous awards for his work and was elected member of the National Academy of Sciences in 2019. And now he will give us a talk about technologies for understanding and repairing the brain. So Ed, the stage is yours. Welcome.
Still probably need to share slides.
Great to be here, yeah, and excited to talk about one of those complex things that we study and that presents huge amounts of challenge, which is the brain. Let me try to share my screen here. Okay, is this working now? Great. So I lead a group at MIT that works on technologies for seeing and repairing the brain. The brain is incredibly complicated. A cubic millimeter of your brain will have 100,000 brain cells connected by 1 billion connections called synapses. And to make it even more complicated, each of those synapses and each of those neurons has chemical and electrical signals that last about 1,000th of a second. So a huge number of processes happen in even a cubic millimeter of the brain every second. And of course, the brain is much larger than a cubic millimeter. One of my dreams is, could we understand the brain to the point where we could simulate decisions, emotions, or other complex brain phenomena in a computer? And ideally, that would be human understandable and biologically realistic. Now, in order for that to happen, we need to develop many technologies for seeing and controlling the brain. Of course, another reason to do this is we want to be able to understand brain diseases as well. Brain diseases affect perhaps over 1 billion people around the world, none of which can be fully cured. And for many diseases like Alzheimer's or stroke, Parkinson's and epilepsy, there's not a lot we can do. So there's great clinical need. So we want to be able to observe what's going on in the brain, map what's going on in the brain, and control it. How do we map the brain? Well, the brain, of course, has many different spatial scales. Brain cells are enormous objects. And as I alluded to earlier, the density of the wiring is nanoscale. If we want to see how the brain is organized, we have to make a map of the brain cells throughout the brain and the molecules throughout those brain cells. Now, I think we've all seen images like this, brain scans. While non-invasive, they're not very spatially precise. Each blob or voxel might have millions or even billions of brain cells in it. At the other extreme are microscopes, but even they struggle to image nanoscale things. So in our group, we often try to do the opposite of what people have done in the past. We wondered, what if we could chemically weave a dense spiderweb-like mesh of swallowable polymer, basically the stuff in baby diapers, inside brain cells and outside brain cells, in between biomolecules and around biomolecules. Do it just right, add water so that the baby diaper material swells, and could we make the brain physically larger enough that you can make a map of the brain? So about a half decade ago, we showed that we could do that. Panel B shows a small piece of the mouse brain. Panel C, the same piece of mouse brain, about a day and a half later. We permeated it with a dense mesh of polymer before we expanded it, and then after we added water, we could blow the brain up to make it much larger. Now we can express glowing proteins, like from jellyfish or corals, in brain cells using gene therapy vectors. Then do the baby diaper chemical synthesis, add water, and blow it up. If you image a piece of the color-coded brain with a regular microscope, you get an image like at the top middle, looks fuzzy. But if you expand it first, you get something like in the upper right, much more detailed wiring. So my dream is maybe we could actually make maps of the brain so detailed, the molecules throughout brain cells and the brain cells throughout networks, that maybe we could try to simulate behaviors, decisions, and emotions. Now, an interesting thing about building a technology for confronting the brain is that it often helps with other problems. And after we showed the world that we could expand the brain, many people came to us and said, "Can you help with my problem too?" It turns out that many of the leading causes of death, if you diagnose it earlier, you can save more lives. For example, if you catch a cancer before it spreads. So we started expanding cancer biopsies and wondered, could we help make the early, subtle changes into big, obvious ones? Early in a disease, after all, it's hard to see what's there. If we could expand the biopsy and physically make the small changes large, maybe we could help people diagnose diseases better. And that's something that now we're actively exploring with many doctors expanding different kinds of tissues and biopsies to see if the early small changes in a disease can be made larger. This, by the way, illustrates a kind of pattern that our group often performs, which I call invent, discover, design. We invented a tool, the expansion method, that lets you see nanoscale tiny things with regular microscopes. We are helping people make discoveries, like the fact that you could see an early stage of a tumor. And then finally, we designed practical strategies for getting them out into the world. Now, of course, one of the obvious limitations of expansion is that it doesn't work on living things. And so in the second part of the talk, I want to talk about a strategy for controlling brain activity while it's alive. The basic idea, which we call optogenetics, is basically the idea that we could control the electrical activity of brain cells by putting little solar panels in them, molecules that convert light into electrical energy. Then you can bring light into the brain. That's not so hard because the brain doesn't feel pain. You can bring an optical into the brain and activate the brain cell. Turns out, all over the tree of life, you can find molecules from microbes that convert light into electrical signals. Light-driven proton pumps, like the one shown on the left, light-driven chloride pumps, like those in the middle, light-driven ion channels, like those shown on the right. These are proteins found in single-celled microbes all over the Earth. They're used as primitive photosynthetic or photosensory molecules. They're encoded by genes. If we put the genes into brain cells using commonly available gene therapy strategies, then we can shine light on the brain cells and pump or transport ions, charged particles, from one side of the neural cell to the other. Then we can turn neurons off and cancel out their activity. That's what the light-driven proton pumps and chloride pumps enable. Or we can activate the neurons and turn them on. By deleting neurons momentarily, you can figure out what they're needed for and turn off pathological activity. By activating neurons, you can figure out what they can drive and activate potentially healthy activity. That's the invention. Now, what about the discovery? Well, thousands of scientists, just as with expansion, are using optogenetics to study the brain. I'll give you one example from Li Wei-Sai's group at MIT. She used our light activator to show she could drive the mouse brain at a specific frequency, 40 times a second. And these are mice engineered to get Alzheimer's-like symptoms. Could you treat Alzheimer's with a brainwave? Amazingly, these mice got better when she drove the brains 40 times a second. Now, that's the discovery. Is there a design that could be brought into the world? And under her leadership, the collaborative went on to show that you could drive these same brainwaves by basically having the mice watch a movie, show it a flickering light, and give it a clicking sound. And so that was also able to help the mice get better. So now there are clinical trials that are ongoing with actual Alzheimer's patients asking, could you use a movie to treat Alzheimer's disease? I think of optogenetics as a tool for science, but some of you may have seen that a few weeks ago it was announced that a European team has shown some success in directly using optogenetics in human patients with blindness. These are patients who've lost their photoreceptor cells in the eye, but by putting these photoreceptors that our group discovered, like a molecule called crimson, for example, into the eye of a blind patient, they could see again. The vision is not perfect, but it's early days, and my hope is that the partial restoration of vision that's been shown in a patient in a trial that José Alain Sahel and Bolton Ruska and others have overseen will potentially enable even better vision in the time to come. So these are two examples that I want to tell you about today where we invented a tool to show people science they couldn't see before. People went on to make discoveries using the tools. And finally, there are practical designs that can be brought into the world to help people. But back to the original prompt. How can we make a model of the brain that allows us to potentially try to simulate decisions or emotions? And furthermore, can we make maps of the brain that help us systematically understand brain diseases? Those are outstanding challenges, but one of my hopes is that we could try to make maps of small brains relatively soon. Imagine if we could look at brain activity in a small organism like a worm or a fish, which is now being done by many people. We can use the optogenetics to perturb the activity, and at the end of the day, we can expand the brain and make a map. Can we bring those data sets together and make a simulation of the brain? My hope is that this will become possible in the years to come, starting with small brains and potentially being scaled up over time. This could have huge impact in AI. Could help us understand something about the human condition. What is a thought, really? What is happening in our brain when we have an emotion? I'll end there, but I just wanted to note that this is a very large collaborative project with many people, both in our group and our alumni at the top, and an even longer list of collaborators all over the world, shown in the middle. And with that, I'd be happy to take questions.
Yeah, thank you very much for the amazing talk. We have quite a few interesting questions from the audience. And let's start with the one from Anders Sandberg, who is asking, could we tell synaptic strength from the expanded version of the synapse?
Great question. So there's when we expand the brain, we're expanding all the molecules away from each other. So it looks to the eye like you zoomed in with a microscope. And that's one reason why it's very popular. Already over 250 papers doing expansion have already come out. Now, can you take the image and correlate that with synaptic strength? Well, to be honest, nobody's really proven that there's a strict linkage between those in any full set of synapses in a brain. In fact, we don't even know how many cell types there are in most species. So that would be a goal to see if we can use our imaging method or any imaging method to infer synaptic strength. And synaptic strength is only the beginning. You also want to know where the ion channels are, right? Some cells might be very excitable. Some might be very quiet. Give it the same synaptic input, and two cells might be doing completely different things. So hopefully we can start to map out the strategies all over the brain of how brain cells communicate and compute, and that could be quite powerful.
Okay, nice, thank you. There is another question from Anna Riznavel. No, oh my gosh, I'm so sorry. How reliable is the mapping of the brain? The function of different parts are known to change when we age or when we suffer from injuries. How reliable is that?
Many people are using expansion to look at different problems like aging or injuries. I'll give you one example that we also did with Li Wei Sai, the person who pioneered the Alzheimer's movies that I mentioned earlier. What they are finding with our technology of expansion is that when we expand an Alzheimer's model brain, we can actually map pathology and reveal patterns of protein organization that have not been seen before. And so my hope is that indeed the expansion process will make brain mapping very reliable to look at these changes that are due to aging or after injuries.
Yeah, that'd be really great if it was the case. Meanwhile, the next question from Nigel Visser. Doesn't this kind of optical activation method only work on the surface area, or do you use a fiber optic inspection tool at certain places?
So you can do both. You can deliver red light, which goes deeper in the body because it's not absorbed by the blood. That's why blood looks red. And we and others have developed less invasive strategies for brain activation. In fact, the molecule that I mentioned, which is helping restore partial vision in human patients, is a red light activated molecule to help get the light in. You can also bring a fiber optic cable into the brain, and many people are doing that as well in animals. That has not yet been done in humans. But for example, in the example where Li Wei's group discovered a brainwave that could help with Alzheimer's, that was initially done by putting the gene for our molecule into the brain and then bringing a fiber optic into the brain aimed at those cells.
Mm-hmm. Okay, thank you very much for your answers and for the presentation. We'll continue with more questions during the panel, but now it's time for the